Now I'd like to introduce our featured presenter today, Dr. Lucia Aronica. Lucia is a lecturer at the Stanford Prevention Research Center and primary instructor of the Epigenetics and Microbiomics module of the Stanford Genomic Certificate at the Stanford Center for Professional Development. Lucia is currently leading the epigenetic analysis of the Stanford Diet Fit study by Professor Christopher Gardner, the largest randomized clinical trial ever undertaken to compare low-carb versus low-fat diets for the design of personalized weight loss strategies. The focus of her research is investigating how diet affects the epigenome and whether we can use epigenetic biomarkers to design personalized weight loss plans. Lucia also serves as an advisor for companies active in the personal genomics and precision health field. Lucia received her PhD from the University of Vienna and has research experience from the University of Oxford, University of Southern California, and University Federico II of Naples. She has published research papers in top-ranked peer-reviewed journals such as Cell, Genes, and Development, and the EMBO Journal. And with that, let's turn it over to Lucia.
So thank you very much for having me here. It's a pleasure to talk today about unlocking your genetic potential using the science of epigenetics. And this is my mission is using science to live a healthier, happier life and helping people achieving this. You can follow us at EpiWellness and find more information at draronica.com. I do this through my research and teaching. If my research could be a painting, it would be this painting here, and not because the author, Giuseppe Arciboldo, was Italian as I am. The reason is that Giuseppe seemed to know that food shapes our biology, and it does so through two processes that are the cornerstone of my research here at Stanford: Nutrigenetics and Nutrigenomics. Nutrigenetics describes how genes can affect our response to diet, whereas Nutrigenomics describes how diet can affect our gene expression. So this is like a give and take, a sort of conversation. This is my main research interest here at Stanford. But I also teach several courses at Stanford in person and online. Yes, you can get notified about new course launches at draronica.com. My research is the driving engine of my teaching, but my teaching on the other side is the fuel of my research. I get a lot of motivation from teaching, and many students tell me that they come out of my courses empowered to use science to improve their health. And I hope that today I can give you a taste of that, and you can take home some messages, actionable information to improve your health. So today we will learn about what is epigenetic and how it is different from genetics, how diet can impact our epigenetics and health, and how epigenetics can help us take ownership on our health. So, understanding the difference between epigenetics and genetics is especially important now that there are several companies using genetic testing to predict everything about us, what we should eat, where, and even what kind of music should we listen to. So all these companies work in the space of precision health, which is a new and revolutionary healthcare approach aiming at predicting, preventing, and curing disease precisely by using biological markers that are unique so they're really unique for people, personalized characteristics. And these biomarkers are genomics, epigenomics, exposomics, so the entire set of environmental factors we are exposed to, metabolomics, the metabolites in our body, proteomics, all the proteins in our cells and tissues, and the microbiome, the universe of microbes within us and on us. But most of the companies in the precision health space are now using only genomics, but we know that the other omics sciences, they're called lifestyle omics, are actually responsive to what we do, to our environment, our lifestyle, and therefore are of particular interest in the field of preventative medicine and lifestyle medicine. And they can also enhance the predictive power of genomics markers when measured alone. So why do we need to integrate genetic testing with lifestyle omics markers for precise health predictions? Let's have a genetic refresher to, in order to answer this question. Our DNA is made of four building blocks for nucleotides, ATGC, that provide the information for building the sequence of the amino acids in the proteins that build up our cells and tissues. And a multi-billion-dollar project running from the 90s until 2003, the Human Genome Project, aimed at sequencing the entire sequence of the human DNA, so the human genome. There was a lot of hope behind this project. I was one of the people very excited about this project. Actually, I started my university studies in 2000, and this was one reason for me to decide to pursue studies in molecular biology and genomics. And scientists were literally betting on how many genes will turn up when the human genome will be finally mapped. But the human genome, as you may know, produced some shocking surprises. The first shocking surprise was that we humans have actually the same, more or less the same number of genes as simple model systems such as mice, fruit flies, and worms, and 50 times fewer than an onion. The second shocking surprise was that genes account for only 2% of our DNA. And if that weren't enough to hurt our genetic egos, there is a third shocking surprise that actually your DNA is 99.9 similar to any other person's DNA. So technically speaking, our DNA can explain only 0.1% of what makes us unique. And therefore is perhaps not surprising that also my genetic report was not very accurate in predicting my hair type. So according to my genetic report, I'm 72% likely to have straight hair. And for the same reason, we were not surprised when we received the genomic results for our study, the Diet Fit study. This is a study by Professor Christopher Gardner with 609 overweight and obese men and women that are randomized to either low carb or low fat diet that they follow for 1 year. We are measuring genomics, epigenomics, microbiomics, proteomic markers for our study. And we published the results for the genetic part, and we actually could not replicate some genetic associations that had been published previously, associating 3 genes in the glucose and fat metabolism with the prediction of diet response, either low carb or low fat. And that's why we are now focusing on epigenetics, and we are measuring DNA methylation, looking for epigenetic biomarkers of diet response. So what is epigenetics? You may remember the 2008 cover of the Time magazine stating that the new science of epigenetics reveals how the choices you make can change your genes and those of your kids. So epigenetics, to understand epigenetics, let's just describe the meaning of the word that comes from the Greek language, where epi means above, on the top. So if your DNA is your genome, then your epigenome is actually a set of molecular markers, chemical groups that are on the top of your DNA and can switch genes on and off. Just like a dimmer switch can modulate lights up and down in a room. And this process of turning genes on and off can explain why we have different types of cells in our body: liver cells, neurons, fat cells, all told more than 200 types of cells, that all shares the same DNA, the same information. So how is this possible? Well, think of your DNA, your genome, as hardware. Then the epigenome is software that can turn genes on and off, specifically in different cells. So each cell will turn off a specific set of genes and turn on only those genes that are required for its identity and function. To understand the software, we need to understand 2 fundamental properties of the epigenome: epigenetic flexibility and memory. So unlike genetic sequence, our epigenome is flexible and dynamically responds to lifestyle factors. This is this happens already very early in life. So when the egg and sperm fuse together, actually they erase almost all the epigenetic marks, giving rise to embryonic stem cells. These stem cells can become any type of cell because they don't have epigenetic markers to instruct the cell about which genes to turn on and off. But when during embryonic fetal development these cells do acquire epigenetic marks, then they do start to differentiate in the different types of cells that make up our body. And as you can imagine, embryonic and fetal development are time windows that are particularly sensitive to lifestyle factors, and this is a reason why pregnant women should be especially careful of their lifestyle, because this can have a huge impact on the epigenome of the developing fetus. There are many lifestyle factors affecting the epigenome: diet, pollutants, stress, drugs, chemicals, additives, and the way these lifestyle factors can impact our epigenome and gene expression is by sending signals to enzymes, proteins that either write, erase, or read epigenetic modifications on our genes, thus affecting gene expression. Therefore, you can think of the epigenome as a set of sticky notes placed on your genes that can store an epigenetic molecular memory that is called epigenetic memory of your lifestyle exposures. The delicate balance between flexibility and molecular stability is what makes it possible for our cells to store an epigenetic memory. Epigenetic marks are flexibly modified by lifestyle factors, but they're stable through cell division and can be therefore passed to the daughter cells. And this makes epigenetic memory possible. Now, the nodes, the markers that are written before our birth tend to be written in pen, so they tend to be permanent, whereas the nodes that are taken after our birth are potentially reversible, as if written with a pencil. And both pen and pencil marks can affect our health and predisposition for diseases. So while genetic factors can contribute to our traits through a process of hardwiring, epigenetic markers do so by a process of soft wiring that is dynamically influenced by our environment and lifestyle. And that's why, for example, identical twins that have exactly the same DNA can, with time, not only look different, but also develop different predisposition to disease. Now, there have been 1,000 studies on the impact of diet on our epigenetics. Actually, epigenetics is an opportunity for us to look at diet from a new perspective. Food is not only calories, it's information. It's the most potent signal, powerful signals to our genes. And it can actually modulate several of those enzymes that can write, erase, and read epigenetic modifications. One of the most famous studies in animal around diet and epigenome is a study coming from a bee hive. So queen bees are much larger than other bees in a beehive that are called worker bees. They also live longer and they are fertile. They can lay eggs, whereas worker bees cannot. Now, what is surprising is that queen bees and worker bees are genetically identical. And what is even more surprising is that the only difference is that only queen bees are fed royal jelly, a protein-rich substance that is actually secreted by worker bees, and this tells a lot about fairness in nature, that can actually turn on specific genes that make the queen become a queen. But before you run to your local store and buy royal jelly, I have to tell you that this doesn't seem to work for humans. We do have nutrients that we know are acting through an epigenetic mechanism, and we are discovering every day new nutrients in this category. I call these nutrients epinutrients. These are our royal jelly. They nourish our epigenome and unlock our full potential. Here is a table with some of these epinutrients. So the one here on the left in orange are nutrients that can actually provide the building blocks for making the switches, the epigenetic switches. These include methionine, folate, vitamin B12, B6, choline, betaine, and the nutrients on the right, this one in violet, they can actually modulate the activity of those writers, erasers, and readers, and so affect our epigenome. These include many polyphenols, such as, for example, those from green tea, resveratrol from red wine, some flavonoids in soy, genistein, sulforaphane, a very famous epinutrient found in all cruciferous vegetables and broccoli, butyrate, a very interesting nutrient that is actually produced in the gut when dietary fiber is fermented. And so it's also heavily dependent on our microbiome. And then diallyl sulfide or DADS from garlic. So all these studies are showing that we are what we eat. But there are even studies that suggest that we may actually be what our parents and grandparents ate. One of these studies is again an animal study, and these two mice here are very famous. As you can see, one is yellow and fat, and the other is brown and lean. And again, surprisingly, these mice are genetically identical. They both have a gene called the agouti viable yellow allele, or AVY, that genetically programs these mice to become fat, develop yellow fur, and develop later in life diabetes. But the AVI gene is turned on only in the yellow fat mouse. Why? Actually, this is because of the diet of the mother of these mice. So for the yellow mouse, the mother of the yellow mouse had a diet that was poor in epinutrients. And for these reasons, her daughter couldn't build the building blocks of the epigenome and turn off the AVY allele during embryonic development. Whereas the mother of the lean brown mouse was behaving well and eating all these epinutrients, was fed the diet, was supplemented with those nutrients. And as a result, her daughter could turn off the AVY gene. But what about humans? Is there some evidence that we could also be what our parents and grandparents ate? Well, there is evidence, not from an experiment, but from a historic tragedy. This was the Dutch hunger winter during World War II. There was a food embargo in Holland, and as a result, more than 20,000 people died. As you can imagine, the mothers, the babies born during the famine were underweight and had a lot of health issues. And this is not surprising. But what is surprising is that when these babies became adults, they actually developed, they were more prone to develop diabetes and become obese. And what is even more surprising is that the children of the famine babies had also a higher risk of becoming obese and developing metabolic disease, although they were never been exposed to the famine. So recently, scientists found out that actually the famine babies had persistent epigenetic changes, especially in some genes regulating metabolism, glucose metabolism and fat metabolism, which suggests that there were some epigenetic marks written in pen during the famine that persisted later in life and had long-term consequences on the metabolic health of these people. And but not only the diet of the mother is important, also the diet of fathers. And this is a recent study showing that actually the epigenetic marks in the sperm cells of obese people are dramatically different from those found in lean people, and they are found in genes that regulate brain function and appetite, which makes sense from a mechanistically point of view to explain the problem with obesity and the metabolic disease. Even more surprising, when these people, the obese people, go through bariatric surgery and lose weight, also these epigenetic markers reverse from the obese type to the lean type. So this suggests that actually the obesity associated epigenetic markers in sperm cells may actually be written with a pencil. And this brings us to the most important topic of this lecture. We want to use epigenetics to live a healthier, happier life and empower us to take ownership on our genes and our health. To do so, we should move from a paradigm of fear of disease to one of opportunity for health. Just think about your genes as the starting line in your health journey. The good and bad predispositions you were born with, and the lifestyle omics are your path to health. Really the biology that you build every day by making your lifestyle choices. So I hope that you can embrace now why lifestyle omics matter. And I will show you one last example from recent studies in which researchers identified a polygenic score able to predict the risk of coronary artery calcification, coronary artery disease in people. And then they looked at how these people could actually predict disease in 3 different categories of people. Some of them had a favorable lifestyle with exercise and healthy food. Some of them an intermediate lifestyle here in gray, and then some an unfavorable lifestyle in red. Now, as you can see, actually, even if you have a high genetic risk here on the right in red, actually, and you have a favorable lifestyle in blue, this can cancel out your genetic predisposition. You see, it's the blue bars are the same height in the intermediate and high categories. So it means that basically, if you have a favorable lifestyle here in blue, it really doesn't matter if you are, have a high genetic risk. And this is what I actually hope to have shown to you in this presentation. So I hope that now you can embrace that we are unique in many ways, and 2 of these ways are our genes, the book your parents wrote for you, and the other more interesting book is your lifestyle, the book you write every day and can impact your lifestyle omics. And remember that diet is one of the most powerful epigenetic pencils you have in your hands. And with this, I would like to thank you for your attention. And yes, please follow us at EpiWellness.
Thank you very much, Lucia, for this very informative presentation. And now we will move on to answering your questions. The first question here is, what are some recommended readings or resources to learn more about the topic of epigenetics?
It depends on what level so if you want a very deep dive, I always recommend the books on the book on epigenetics. I think now is the third edition by David Ellis and Thomas Genuine. That is a wonderful book. Otherwise, I do like reading papers, but I know that is a jungle for for most people. I think there are also some websites like What is Epigenetics that can be also very helpful.
So our next question is, how far does the epigenetic flexibility go regarding diet? And does one meal affect as much as lifelong dietary habits?
Oh, I wish I could respond to this question, answer this question with a precise answer. We don't know. Because we don't have studies that have compared one single meal versus a long-term diet exposure. But we do know that actually some epigenetic marks can change very rapidly, such as those that are responsible, for example, to trigger immune responses. We need to switch on and off those genes very, very quickly. So certainly we can predict, it's very likely that even after one meal, there would be some epigenetic changes. The big question is whether these actually matter for your long-term health. It seems that, for example, if you want to reverse some metabolic issues or obesity, those changes require a longer exposure, for example, longer diet intervention. Even the speed of reversal depends on how strong the exposure has been and how long. For example, in case of obesity, how long the exposure to an obesogenic diet has been and how long you have been overweight. There is an hypothesis according to which actually people may have harder time losing weight because of some epigenetic stiffness. So the resistance of some of those pencil marks to actually be raised. So to come back to your question, is really the speed of reversal is really an individual issue.
So this next question is somewhat related, which is, are epinutrients more important during particular parts of a person's life, for example, pregnancy or childhood?
Yes, yes. As I mentioned in my lecture, pregnancy and childhood are especially important because the fetus, during embryonic development, our epigenome is just being established. I describe it as a naked epigenome, or almost naked. Of course, all the life environmental signals in that time window are going to have a bigger impact and are going to be potentially written in pen. I also want to mention that actually it's a good thing that we have markers that are written in pen, because those markers are essential to establish cell identity. Otherwise, we would have our hair turning into a nose or something like that. So it's good that those markers that are established during embryonic development tend to be written in pen. But the downside is that some of the markers that are established, for example, in response to a bad or adverse lifestyle exposure, might as well then end up written in pen.
Okay, so the next question is actually regarding that, which is there a way to turn off the genes that can impact health negatively, for example, the famine genes?
Yeah, again, there is evidence of epigenetic reversal of some disease-associated epigenetic markers. I've shown you the example of the obesity associated markers in the sperm of men, but actually also smokers. Smoking associated epigenetic markers are exceptionally very well studied. Actually researchers have shown that those markers can be reversed after 16 years. So after quitting with smoking. So there is evidence. Again, the speed of reversal depends on the exposure and the type of exposure, and is a person on the person.
Okay. Is it possible to discriminate a stop or go signal through CRISPR-Cas9 or similar editing gene system?
Yeah, so this is more like a genetic question, right? So you are here. Your suggestion is to manipulate actually the DNA sequence. We cannot do that now. I mean, there is ongoing research in animal to achieve that. But I'm a believer of lifestyle medicine intervention. So rather than changing the DNA, changing the way it works.
The next question is, I teach health and PE to high school students. We're starting nutrition soon. Any ideas or hints to change their poor diet for 14 to 16-year-olds?
Wow. So this is also more than a genomic question, more a diet question. So the question, what do we know about what is the most impactful intervention that we can do to improve our health and perhaps our epigenetics? Well, for sure, there is a consensus on the reduction of refined grains and added sugars. There are in the epigenetic field some publications suggesting that sugar can actually trigger epigenetic modifications that link metabolic disease to other diseases that are associated to obesity, including cancer. What we know from dietary recommendation has also a more profound implication in our deep biology and epigenetics. Reduction of refined grains and sugars is for sure important. Then a diet that is rich in fiber to promote the microbiome. There is also now new research showing actually a crosstalk between our microbiome and our epigenetics. So those two are two areas of that are very important now. And then, of course, the third is quality of food, good quality of fats and carbohydrates. So carbohydrates primarily from vegetables and fats also from natural fats, not hydrogenated industrial fats.
Okay, so you mentioned the epigenomic role of the father influences brain function and appetite control genes, but what about the mother? And to what extent can an unhealthy mother help reverse any potential damage she might have superimposed on her unborn fetus?
Yeah, so regarding the effects, so the genes that showed differential epigenetic modifications and expression in the famine babies, so for the one that were modified as a consequence of the mother exposures, those genes were especially metabolic genes, so genes involved in the glucose and fat metabolism. As I mentioned, the research about how to reverse those marks is ongoing. There is hope. Actually, epigenetics is the science of hope. Whereas genetics, genetic determinism, may induce people to think, okay, I'm stuck with my genetics, there's nothing I can do. Epigenetics give us a tool to actually take action on our health and improve our health. There is research, as I mentioned, showing that many of those marks can be reversed. Even those in the fetus are not all written in pen. Not all the notes written during embryonic development are written in pen. So I think it's really important to act as soon as possible in case of children that have been exposed to adverse environmental exposure during a pregnancy. It's important to act as soon as possible, because also childhood is a very sensitive time window for the epigenome. And by doing so, and focusing on all the environmental factors that we know can benefit the epigenome. Diet is one of them, but also there's emotional health and environmental health, exposure to chemicals. I'm sure you can minimize the effects of a bad exposure during a pregnancy and maximize the likelihood of a thriving life for your children.
Okay. So there was a couple of additional questions here, and we have a little bit more time. And so someone had asked, they had their entire genome mapped. What should they be asking when they see the results?
Wow, this is a big question. So it's actually very exciting, and now more companies are offering the mapping of the entire genome. As you may know, there is a difference between mapping the entire genome or genotyping through a service like, for example, 23andMe or other genetic testing companies. But now there is also the question how to use that information. This is a big question here. Genetic testing can be a double-edged sword. So on one side can actually empower people to take action of their health. You can say, okay, I have this predisposition to absorb less folate from my diet, so I will eat more of that. But on the other side, this can also trigger some nocebo effect. Like, for example, people can feel that they have bad genes and there's nothing they can do. There is actually an interesting study published by Stanford researchers a couple of months ago, showing that people that received fake genetic scores, predicting that they were going to perform poorly on a treadmill, then actually when they did, and they did the treadmill test, and they did perform poorly, but those were fake genetic scores. So my point here is, when you receive this genetic information, there are some genes that you can look at. There are a few genes that are really backed up by convincing evidence. And these are especially genes in the regulating our response to micronutrients like folate and also some other vitamins, vitamin D, and also the omega-3. So there's some people, for example, that have some genetic polymorphisms that make us less efficient at converting plant omega-3s to the biologically active EPA and DHA. So there is evidence that there are genetic polymorphisms that are backed by evidence, but you should always be aware that those our complex traits, all our traits, even our response to food, are actually regulated by 100,000 genes. And that's why it's very difficult to predict them precisely only by genetic testing. One example is height. Height is highly genetic, so is a genetic trait, but it can be explained like from 80 to 90% by genes. But because it's affected by more than 100,000 genes, there is actually now no genetic test that is going to able to predict height. So my point here is, yes, go look at some, there are areas that you can look at, like micronutrients, omega-3 response, also methylation pathways, cardiometabolic health. But please be aware that those are only predictions with low predictive power, because complex traits are affected by 1,000 genes. And hopefully one day, when we will be able to integrate those genetic predictions with other multi-omics markers that I showed you, then we'll be able to enhance the predictivity of those markers and be more confident about their meaning for our health.
Thank you. All right, well, unfortunately, we're getting close to the hour, so we'll have to wrap up the Q&A session. Thank you very much, Dr. Aronica, for sharing your knowledge with our participants today. For those of you who have indicated an interest in the course, our team will be reaching out with more information, and you can always visit us at geneticscertificate.stanford.edu. As a final reminder, the recording of this webinar will be emailed to you within a week. Thank you all for joining us today, and have a great rest of your week.